Right, so this is the second part of chapter 2. We're going to cover the processes of exocytosis and endocytosis, talk about coated vesicles in membrane transport, and finish up with 2 organelles involved in digestion of materials from the cell, the lysosomes and the peroxisomes. So let's start with exocytosis. So exocytosis is also known as secretion because the cell is putting molecules out to the outside of the cell, and these are larger molecules. So this is not stuff that would be going through active transport or facilitated diffusion or diffusion. These are larger molecules, and this is also called bulk transport, which means instead of moving a couple molecules at a time, you're moving a whole bunch. And a really good example of this is the production of antibodies. So we draw antibodies like this. And your B cells, which are part of your immune system, will produce these protein antibodies in the cell and then mass secrete them through exocytosis out into your lymph tissues, into your blood, so that those antibodies can circulate and find foreign invaders. The key to exocytosis and endocytosis is membrane fusion. So we talked about, in the first part of this chapter, the movement of vesicles from the ER to the Golgi to the plasma membrane to other organelles, and it's all made possible because membranes are membranes are membranes. And so these phospholipid bilayers can fuse together and become one. Exocytosis has several different types of secretion. There's constitutive, which just means constant. So or as it says here, unregulated. So the example they give in your textbook is mucus. And mucus is this thick fluid that contains a bunch of enzymes, such as lysozymes, things that can destroy foreign molecules. Mucus contains antibodies, glycoproteins, inorganic salts. So it makes this whole package here of mucus that it constantly secretes out through our nose, our lungs, our respiratory tract, our GI tract, our urogenital tract, and even your eyeballs. So you are constantly secreting liquid, not necessarily thick mucus, but in your eyes to keep them moist. So constitutive secretion just means unregulated all the time going. And that's in contrast to regulated secretion. In regulated secretion, the contents of the secretory vesicle are not released until there's an extracellular signal. And we don't really talk about signal transduction until the last section of this class, which is shown right here. But you can imagine regulated means something outside of the cell is signaling the cell to go ahead and release the contents. So that's regulated. The third type is polarized. And when we talked about cell junctions, we mentioned that some cells have a specific apical and basolateral layer, or ends to it. And so cells that have this polarity, have different ends to them, can do polarized secretion. So it's not that the secretion can happen anywhere out of the cell. It happens at one end or the other. And the example that we will keep coming back to is in neurocells or nerve cells. And right here, the secretion of neurotransmitters goes in a single direction. So it goes from the axon of one to the dendrite, or the axon, or sometimes the somatic cell body of another one. But the secretion is always this direction. It's always at this end of the cell. So that's polarized. And what you will learn is that neurotransmitters are both regulated and polarized secretion. So keep those terms in the back of your head. We'll use them later this semester. I put this figure in because I think it's super cool. And next unit, we start talking about the cytoskeleton. And so what I just want you to see from this is that these vesicles in yellow here with their contents, the way they get from one area of the cell to another area is through the cytoskeleton. So the green things are microtubules, which again we will talk about in the next chapter. And this guy kinesin is a motor protein. And so it's really cool. These motor proteins actually kind of walk along the microtubules, and they can carry vesicles from place to place. So this can happen between the ER and the Golgi to the plasma membrane. The other part of this slide that I want you to understand is this docking and fusion. And what I want you to know is that exocytosis, endocytosis, membrane fusion, movement of the vesicles is specific. So I want to talk about Snare hypothesis. And basically what this is telling us is there is specificity in the sorting and trafficking of vesicles. Or I should say sorting and targeting, because they're going to traffic, they're going to move. Okay. And we're looking specifically at exocytosis right now, but you can see that this can happen between, again, the ER and the Golgi and other endosomes, lysosomes, things like that. So there are a couple important components that provide this specificity. And the first one are these Snare proteins. Okay. And there's V-Snare, excuse me, and T-Snare. V-Snare are the proteins on the vesicles, and T-Snares are the proteins on the target. So this diagram is just to show you there are different V-Snares and T-Snares, and they specifically interact with each other to get vesicles to where they are supposed to go. So if we look at the Snare hypothesis, and this is in your textbook as well, excuse me, there are a couple other proteins involved in specificity. So the specificity is via the V and T snares. but also through these proteins called tethering proteins. And when I look for good Snare hypothesis images, a lot of them don't include the tethering proteins, so I added them in here. And the only reason they're disappearing here is just to kind of make the picture more simple, but they don't disappear because they are on that target membrane. So tethering proteins are on the target membrane, and they reach long distances. So they were actually the first to be able to interact with the vesicle. So it's like extending your hands to reach for a ball that's being thrown to you. Once you catch that ball, you bring it in closer. Okay, and so that's what's happening here. The tethering proteins are grabbing that vesicle. They are recognizing something specific. They are bringing it down so that eventually the V-snare and T-snares can interact. The third part of our specificity is this Rab GTPase. And you're going to see lots of GTPases as we talk more about these internal kind of movements in the cell, and they use GTP for their energy instead of ATP. So the Rab GTPases also have some specificity, and what's happening right here is the Snare proteins zip together. So they have alpha helices. Zip together, sorry. Together. They have these alpha helices that start to interact. So it's kind of like putting two springs together. And that zip together helps lock them in place. Okay. And then you have GTP hydrolysis that also is important for locking these proteins together. So this takes energy to get this vesicle in place for membrane fusion. Then we have some proteins that are really not specific, the SNAPs and NSF, and their role is to, one, help the membranes fuse, right. So just to make sure you understand the orientation, the green down here is the target membrane, the green circle is the vesicle, right. And just like I showed with the exocytosis picture, the vesicle and the membrane can fuse together and release the contents. So SNAPs and NSFs come in. They're non-specific. They help the membrane fuse, and they also release the snares that are bound. So the snare interactions, and they release the GTPase. And as this diagram shows, it's believed that ATP energy is required for this to work. So by breaking this interaction here, the membranes can now fuse and the contents are released. This is just another diagram showing the snare interactions. Again, here's those alpha helices. So the blue is the T snare. And the orange here is the V snare, and you can see how the coils kind of wrap around each other. Here's one of the SNAP proteins that's going to help break this interaction so that the membranes can come together and fuse. Here's our Rab GTPase. And again, the reason I add this one in is, remember this term synaptotagmin, sorry, I always say that one wrong, synaptotagmin, and calcium. Because again, when we get to the last unit, talk about neurotransmission, calcium is an important regulator of exocytosis and neurotransmission. So this is an example of regulated exocytosis, and the next few chapters, especially when we start talking about muscles and cytoskeleton, and then this, we're going to talk about calcium ions a lot because they're important regulators of things happening in the cell. So more information than you need, but I like to show you that there's lots of connections within this class. All right, so we've talked about exocytosis, releasing things. Now let's talk about endocytosis. So this is where we're bringing things into the cell. Okay. And we have a couple different types. Phagocytosis is large, solid things like eating a bacteria if you're a macrophage, or taking in large globs of starch. So phagocytosis means to eat. Pinot or pinocytosis, I call it Pinot because then it reminds me of Pinot Noir wine. That reminds me it's to drink. So this is bringing in soluble molecules. So it's not just water, liquid, it's everything dissolved in that liquid. So our cells can get some nutrients this way. Sorry. And the third type that we're going to focus on is receptor-mediated endocytosis. And this has a degree of specificity. Because you're bringing in something that specifically can bind to the receptor on the plasma membrane. What is common about all of these is that it's the plasma membrane that is folding in and pinching off and creating an endocytic vesicle. And even though this says vacuole, we're not getting into such details. We're going to call it vesicles. So endocytic vesicles. Vesicles formed by endocytosis. All right, like I said, we're going to focus on receptor-mediated endocytosis. And receptor-mediated endocytosis involves a protein called clathrin. So clathrin is this triskelion that will aggregate into this 3D vesicle structure. So clathrin and the coat proteins we'll talk about in just a little bit are really important for actually building the scaffolding to make a vesicle, right? So if you think about it, you need some kind of structure to grab that membrane and either bring it in, well, yes, and bring it in. Let's not say either. Grab that membrane, wrap it around something, and bring it in. And that's where clathrin comes into play. So this is specific endocytosis because it uses receptor ligand interactions. So this little blue molecule is called the ligand, and it's whatever can specifically bind the receptor. What's important with clathrin is you also have adapter proteins, and this gives it a level of specificity. So the adapter proteins are like a bridge between the clathrin and the receptors. You can see the adapter proteins in these little pink ovals. They're binding the receptors, recruiting the clathrin. So they're helping promote assembly of this clathrin coat. So as the clathrin forms this lattice structure from the triskelions, it will start bringing in the membrane and start making a vesicle. Eventually, the clathrin will come off and be recycled. Just making sure I've covered everything on my notes. Oh, I made a note to myself. So I have posted a bunch of videos that can show the formation of these a lot better than these static pictures. So there's some animations that I suggest you watch. Another component that's important in receptor-mediated endocytosis is dynamin. And dynamin is also a GTPase. So this is providing energy. And what dynamin does, as you can see, it encircles, it makes a ring around the vesicle and the plasma membrane. And it's like a cinch, it's like a tightening of a belt. So it keeps tightening, 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 tightening until you pinch off that vesicle and you're just left with your clathrin-coated vesicle. As I mentioned, you have to have uncoding. And uncoding not only important for the recycling of clathrin and the adapters, you can also recycle the receptors. But uncoding is essential for membrane fusion. So in order for that vesicle to fuse, perhaps with an early endosome or with the Golgi or wherever it's destined, you have to uncode it. So now you're back to membrane for fusion. Uncoding is thought to be very ATP intensive. So I think your book said like 3 ATP molecules per triskelion, so really expensive energy. Or a very energy rich process. One of the reasons I find receptor-mediated endocytosis so interesting is that this is a way a lot of viruses use the cell to actually trick them to taking them in. So there are some viruses that bind to receptors on our cells. So these are receptors that we normally use for other processes. Okay. And the viruses have evolved to bind to these receptors and trigger endocytosis. And then what's really cool is a lot of these endocytic vesicles then will fuse and become a lysosome, and you get digestion. And we'll go over lysosomes in just a little bit. What's interesting about viruses is that they actually thrive, many of them, at that low pH, and it helps open up this capsid protein and allow the virus to actually infect. So viruses will trick our cells into using something that we normally use, receptor-mediated endocytosis. It will bring the virus into the cell, and then the virus escapes from the endosome and causes infection. Something else that's important, and I kind of mentioned this, so I just put another figure, that receptors are recycled. So the cell uses these receptors for receptor-mediated endocytosis, brings them into the cell, and then can put them back to the plasma membrane. Okay. This is also a way to control the amount of receptor. on the cell surface. So the more receptor you have, the more you're going to respond to outside signals. So one way a cell can desensitize to an outside signal is pulling in the receptors through endocytosis, holding on to them, and then moving them back up to the cell membrane when they're ready to have another response. The problem is if you don't do this, if you don't regulate the amount of receptor on the cell surface, then a cell can be overstimulated. So it has so many receptors, it keeps responding to whatever's out in the environment. That could cause things like cancer, overproliferation. So receptor recycling is an important use of endocytosis. There's also this type called transcytosis, and this is where you're moving components from one side of the cell to another, or one region to another. Again, this is really used a lot in cells that make linings, because these cells have an apical side and a basal side. So some of the key types of transcytosis is with antibodies. So antibodies might be floating around in the mucosal surface, like of your gut, and will be taken in and transcytosed through to the submucosal to go around and look for foreign bacteria or viruses. They are using transcytosis for drug delivery. So they're producing a drug that binds to something we have a receptor for, and that's being taken across the membrane. And so this is being taken out of the blood, across the endothelial layer, and here it's targeting tumor cells. So transcytosis just means across, trans means across, and it's a way to move molecules from one side of a cell to another side of a cell. The caveolae here, we're not going to talk about. Your book doesn't talk about it much, talks about it with respect to cholesterol movement. It's another kind of coated vesicle that moves from the plasma membrane, but we're not going to focus on it this semester. Another type of endocytosis is called fluid phase, and this is really like pinocytosis. And the reason I separate it out is I want you to understand that it's important for compensating for cell surface. For an increase in cell surface or cell surface membrane. Go away. Go away. Sorry. So if you have a cell, I'm having my cell here, and it's doing a lot of exocytosis. The cell is going to grow in membrane size, right? So if I made this super thick, and then I made, here's the vesicle, and here's the vesicle, and here's the vesicle. So I'm increasing this surface membrane because I'm putting all these vesicles up there by exocytosis. And so fluid phase, pinocytosis or endocytosis, can now bring some of that membrane back. And it's not necessarily that specific membrane, but it's to keep the cell surface size correct. You don't want this super big cell, because remember, if you have a really big surface area and a really big volume, if you're increasing the size of your cell, you're not going to be able to make things diffuse as well. So you want to keep that nice surface area to volume ratio. All right, there are other coated vesicles besides clathrin. Table 12-2 in your book is a great one to keep on hand, unless you memorize all these guys. I like this image just because it kind of summarizes. We've talked about clathrin, and remember, clathrin can go from plasma membrane to endosomes, right? Endocytosis, or clathrin can also go from the trans-Golgi network to the endosomes. Okay, so they're in red right there. COP1, coat protein 1, goes from the Golgi to the ER, or Golgi to Golgi. Okay. Oops, sorry. So that's shown right here in green. So it can go retrograde, retrograde Golgi to the ER, whereas the COP2 is going ER to Golgi. Okay, so at least it's showing where it starts. And so let's look at these different coated vesicles. It's the same idea. These coat proteins are important for the formation of the vesicles. And then what I want you to see also is very important are GTPases. So ARF and SAR1 are GTPase enzymes giving energy for the formation of these vesicles and the binding. Excuse me. So this image is kind of split in half, COP1 on the right, COP2 on the left. I just want you to see that these are made, these coated vesicles are made of multiple proteins coming together. Sec proteins stand for secretory proteins, so they can be proteins that are helping form the secretory vesicle, or proteins like we saw on the ER translocon, some sec proteins involved in making those pores. Don't forget, whenever you have a GTPase, you're going to have a GEF protein, which is guanine nucleotide exchange factor. So, for instance, ARF here, ARF comes in two forms, ARF GDP and ARF GTP. The ARF GTP is the active form, but once it hydrolyzes GTP to give the energy, then it becomes ARF GDP, and that's when your guanine nucleotide exchange factor comes in and flips out the GDP, replaces it with GTP, so that the ARF can go and work again. So the same with SAR1 and COP2. So remember what guanine nucleotide exchange factor, or GEF, is. Again, you don't need to know all the details, all these different sec proteins. Just trying to show you some different images. The idea is the same. COP2, COP1 work together with their GTPases to form these coated vesicles. It's important for moving, again, from either ER to Golgi, or Golgi to ER, or Golgi to Golgi. And they have some specificity to help target those vesicles. Okay, we're going to move to our last set of topics, which is the lysosomes and the peroxisomes. What I want you to remember from this picture is that the peroxisomes are not part of the endomembrane system. Okay. So they're not formed from the ER to Golgi like these other vesicles. That means that peroxisomes get their proteins through post-translational import. So we'll talk about that in a minute. They're similar, and then they're both for the breakdown of molecules. Okay. So let's talk about lysosomes first. So lysosomes digest macromolecules. This is what you learned in the first 2 weeks of class. So they can digest nucleic acids and proteins and carbs and lipids, and they can take off phosphates and sulfates. So they use something called acid hydrolases. So hydrolase uses water to break things down. Acid means these are only functional at very low pHs. The inside of this membrane is coated with sugars. We think that this sugar coating protects the membranes and any transmembrane proteins from the acid environment and these enzymes, right? So you don't want a lipase chewing up your membrane, or else you're going to release these contents and this acid into your cell, and you're going to cause massive destruction. Not only do they degrade macromolecules, they degrade old organelles. So sometimes you get a damaged organelle or one that's not functioning as well. So it will be taken into a autophagosome and then bound to a lysosome and digested and recycled. So the lysosomes are really good about recycling macromolecules, old organelles, parts of the cell, so that our body can keep, or our cell, I should say, can keep reusing it. It's a very different pH inside the lysosome than outside in the cytosol. So lysosomes have an ATPase hydrogen ion pump that pumps hydrogen ions into the lysosome lumen, the inside of the lysosome, and creates that very low pH, very acidic environment. And that low pH is also good for denaturing molecules. So it starts to kind of break things up, and then it allows these specific enzymes to become active. The last thing that lysosomes are important for digesting is foreign substances. So something that's taken in through phagocytosis will merge with a lysosome and be digested. So your macrophages will take in bacteria and they'll be digested, broken up into smaller parts. So lysosomes are also important for nutrition. So pinocytosis is a way to get macromolecules into the cell that couldn't normally diffuse but are soluble. Lysosomes and phagocytosis are good for getting much larger. And again, it says vacuole. We're not going to go that specific. We'll talk about the vesicles. Here it is digesting a broken organelle. Remember that lysosomes come from the Golgi. It's sometimes, I think, kind of confusing how different parts of the book talk about the formation of lysosomes and different sources. So just remember, you have endosomes, and early endosomes, and late endosomes, and lysosomes. But the key to a lysosome is it's not a mature lysosome until it has that low pH and it's full of acid hydrolases. Okay, that is a true lysosome. We're not going to get into all the variations of how it becomes one. A couple other terms to note. One is that these proteins, lysosomal proteins, have a specific tag, and it's called a mannose, which is a sugar, 6-phosphate tag. So those proteins that are made in the ER, modified through the Golgi with a mannose 6-phosphate tag, will end up in these lysosomal vesicles. At the end, you sometimes are left with indigestible molecules. Okay, stuff that your lysosome just can't break down any further. Sometimes that stuff is secreted out of the cell via exocytosis. Sometimes these lysosomes become what we call residual bodies, where for some reason they start to just accumulate all this stuff that you can't digest. And the older a cell is, the more residual bodies you will find. And we think this has something to do with cell aging, and perhaps a signal for apoptosis, something we haven't talked about yet in this class. Okay. There's one other way residual bodies are used, and in the immune system, oh, that was cool. In the immune system, certain cells of the immune system will take those residual bodies and present them, which means they put them on the outside and they train your immune system, hey, this is foreign crap. You need to make some T cells to respond to it, or you need to make some antibodies to respond to it, because when you see this again, you need to tag it and destroy it. So this is especially in your white blood cells that have taken in, say, a bacterial cell, and they've broken it down as much as they can, recycled what they could, and then the residual body is actually used to present these little pieces that are left over to the immune system to help train the immune system on what is foreign. All right, I think that's all I want to say about lysosomes. Finally, peroxisomes. So peroxisomes are another type of digestive organelle, but they don't use acidic environment, and they digest totally different stuff. The main enzyme that characterizes peroxisomes is the enzyme catalase. Okay. And you may have done this if you've taken microbiology already. We look for catalase positive bacteria, or if you go put hydrogen peroxide, which is an antiseptic, on an open wound, you will see it bubble. So that means there's catalase enzyme there that's breaking down the hydrogen peroxide to water and oxygen, hence bubbles. Hydrogen peroxide is a great disinfectant in the sense that it can cause a lot of damage to proteins and lipids and even DNA. So if you've got an infection, it's a topical way, okay, so on the surface, to try to kill some of that bacteria. Now, a lot of bacteria produce catalase so that the hydrogen peroxide doesn't harm them. And hydrogen peroxide in large doses can be very detrimental to your own cells. So there is some controversy about whether you should or should not use hydrogen peroxide. Another characteristic of peroxisomes is that in certain organisms you can see this urate oxidase crystalline core. Right? So it's this enzyme that crystallizes inside. What's super interesting is this enzyme is not in humans or primates. So non-human primates as well. And so I was like, yeah, well, whatever, who cares? Well, apparently, urate oxidase is found in all other, like, meat-eating organisms. So it's even found in, so what I read was, of all animals that include meat in their diet, humans are the only animal that is unable to break down uric acid, which is what urate oxidase does. Okay? We have the gene, but it's no longer active. So sometime when primates evolved, something happened to that gene. So you can find urate oxidase in bacteria, other mammals, in beans, in legumes, in yeast. And so the question is, why did we lose the function of this? And urate oxidase, like I said, or maybe I didn't say, catalyzes the breakdown of uric acid. And if you have an accumulation of uric acid, you get a disease called gout. So it's interesting that we don't have this functional enzyme, and the idea is that uric acid is an important antioxidant, so it helps get rid of things, not hydrogen peroxide specifically, but oxygens that are free radicals, OH minus, different kinds of reactive oxygen species that we make from metabolism. So maybe we want to keep uric acid around to help get rid of these. Anyways, long story short, you can look at your peroxisome organelles and they do not have this crystalline core. So peroxisomes best known for catalase, breaking down hydrogen peroxide, but they do some other important digestive functions. So for instance, peroxisomes break down long-chain fatty acids and branched fatty acids. So apparently that's something that lysosomes cannot break down. They do it through this thing called beta oxidation. Peroxisomes do a lot of detoxification of free radicals. And free radicals are something with an electron floating around that's very reactive. So again, free radicals can break down your lipids, can break down proteins, can damage your DNA. So we want to have ways to detoxify or break these down. And finally, peroxisomes are also important in metabolizing or breaking down nitrogen-containing compounds. So lysosomes use an acidic environment to break down a lot of macromolecules or organelles that are no longer useful or things brought in by phagocytosis. Peroxisomes are more of the detoxification of these free radicals that can cause harm. Hydrogen peroxide, which can break down into free radicals, fatty acids, and nitrogen-containing compounds. And, because, let's go back here. Just remember that because peroxisomes are not part of the endomembrane system, the proteins for peroxisomes are translated in the cytoplasm or cytosol and then brought to the lysosome by post-translational import. So they're made on a free-floating ribosome, translated, and then imported into the peroxisome. Peroxisomes are also made, and this gets a little confusing. Some places say they're made from vesicles in your Golgi, but then they would be part of the endomembrane system. Some say the membranes are made by the phospholipid exchange proteins, which we learned about last chapter or two. And so what happens with peroxisomes is something called biogenesis, where the peroxisome will actually increase in size and then divide in half. And there's a figure in your textbook that shows this. So, just kind of a little FYI, something I think is pretty interesting. All right, that finishes chapter 12 for us. Thank you.